As I mentioned, I will provide some context to the agricultural sector of South Africa. The South African agriculture is quite diverse, as you can see from this image. This is mainly due to our different climates that we have, which ranges from subtropical to Mediterranean. And this allows for the cultivation of a wide range of products, including wheat, maize, and sugarcane, which is mostly around the northern and eastern parts of the country. We have oilseeds such as sunflower and canola, the citrus fruit, grapes, and various deciduous fruits such as peaches, pears, apples, and plums, just to name a few. And we also have some tropical fruits as well, such as pineapples, bananas, and lychees. And then we have a growing olive industry, as well as our own indigenous teas called rooibos and honeybush. The agricultural sector contributes significantly, obviously, then to the economy of the country, not only through exports, but also through implementation of a circular economy approach across the sector in collaboration with other industries. But one of the main challenges that is faced by the industry is water scarcity. This is then basically require careful management and innovative irrigation practices, as well as an increasing amount of solid waste that is being generated is also a problem, which typically then ends up on landfill at a great cost to the industry. One of our first projects started nearly 20 years ago. It was aimed at addressing the problem related to finding innovative ways to produce clean water from winery wastewater for irrigation. The South African wine industry is one of the top 10 wine producers globally, with the majority of the farms being located in the Western Cape province of South Africa. So you can imagine that with such a large amount of wine being produced, there is a huge problem with winery wastewater. This is a basic overview of the winemaking process. Other steps can also be included, such as clarification and blending. But during this process, you have that the crushing and fermentation vats have to be washed out before it can be reused. And they usually use potable or drinking water, which then result in a wastewater stream that is quite high in organic content, such as ethanol, acetic acid, sugars, and phenolic compounds. And it often contains inorganic salts, such as potassium, sodium, calcium, and magnesium, as well as other reagents that are used for the vat washing. So because of the variation in the use of different types of grapes and the seasonality of the process, the chemical oxygen demand, or COD, is highly variable, ranging from 500 to 4,000 milligrams per liter COD. The solid waste which is generated is called must, can feed into composting, which is typically used on the wine farms, or it is utilized by other companies for extraction of tartaric acid. So this solid waste is actually very well treated. The wastewater, however, can be a problem, especially for smaller cellars, where they have insufficient space for wastewater treatment infrastructure. So the winery effluent at the small cellars are typically used to irrigate fields or pastures. But as you can imagine, using this effluent for water for irrigation causes additional problems. The high level of organics causes anaerobic soils. It is toxic to both plants and microbial communities in the environment, while the inorganic compounds that are present causes the development of sodic swells, where an excess of the exchangeable sodium negatively affects water infiltration and aeration in the soil. So then further downstream, you also have the problem that the effluent can also cause eutrophication of other aquatic systems, which then leads to algal blooms and a toxic environment for vertebrates and invertebrates. There's therefore a need for a simple yet affordable system to treat the winery effluent from small cellars. One way to address this is to ask how nature achieves this. So in collaboration with Biomimicry SA, we approach the problem by asking the relevant questions. So what is the problem? How does nature solve this particular problem? Are there any examples in nature? And can we find the process in nature that achieve reproducible success? Can we then develop a solution based on this natural model? And then does our design emulate nature? In other words, can it adapt and evolve? Is it conducive to life? And is it sustainable? So then, taking these questions into consideration, our team visited various small vineyards in the Western Cape province to see how farmers deal with winery effluent and if a successful process in nature can be found. So on one of the farms, which is called Nietvoerbij, had a natural wetland located next to their processing facility. The winery effluent entered the wetland system as a stream high in organics and inorganics, but showed a reduced levels once through the wetland system, suggesting a natural bioremediation process taking place. But now, unfortunately, not all small vineyards have wetlands on their properties. So how can we mimic this successfully? Based on this observation, we started with setting up biological sand filters using locally available sand to try and mimic the natural wetland system. During this process, the effect of sand physicochemistry, as well as the effect of microbial communities on winery effluent treatment, was monitored. This then reflected both the biotic and abiotic processes that takes place during bioremediation. We also monitored whether organics and inorganics and salts were removed, and we monitored this all by using HPLC and a spectroquant. This then ultimately allowed us to select the optimal sand type and mixture, the load, and the optimal retention time to ensure the removal of harmful substances. The removal efficiency was monitored at the different levels of the particular biological sand filter, so at the inlet and the outlet, as well as the top, middle, and the deep sediments in the biological sand filter to make sure, or so that we can understand what is actually happening within the whole system. The key points highlighted for a successful system included the need to acclimatize the microbial population to the winery wastewater. It was typically a 2-week exposure period that is required. You also have to understand the role of the hydraulic flow and redox status within the system, and the use of a suitable substrate or sand, and then obviously developing a system that not only remove organics, but also salts. Here are some of the examples of results obtained while monitoring the functionality of the biological sand filters. In the first figure, the link between redox status and removal of COD at the different locations, such as the outlets and inlets in the system, can be seen. Within 24 hours of a high COD influent, more than 70% was removed, while in the second image, insights into potential role of microbial communities could be monitored by evaluating the effect of high COD effluent on these communities at the different sites of the system, with the top plot indicating the communities before and the bottom plot after exposure. How the communities are affected can then provide guidance to the development of more resilient communities that acclimatize to a high and variable COD. So ultimately, the work over the years led to the development of a treatment system that could be implemented. This final treatment design, consisting of a dual sand filtration system, was implemented on a small vineyard called Neertlingshof, and it is currently still in place and it's functioning. Some of the design highlights are that it's quite simple to install. As you can see, it's very few components that are required. It's very simple to operate and maintain, and all of the components were available off the shelf, and a locally available sand resulted in ideal flow characteristics, and no energy was required for the operation of the system since it's entirely run on solar-powered pump and gravity. The system was also monitored for a 2-year period, going through 2 different crush seasons. The specific highlights include the fact that the removal of the organics made the treated effluent compliant with legislative standards, and could be used for irrigation. The system also adapted quite well to a variable influence, so whether or not white grapes or red grapes were crushed. And the removal performance improved over time. There was no sign of clogging, and there was an effective salt removal that took place. And this was all thanks to the abiotic processes that happened within the treatment system. The work presented here, I mean, it's just in a few slides here. It actually required extensive collaborations, bringing together microbial ecologists, biotechnologists, engineers, and the wine farming community to produce this final product for the generation of clean water. Here are just some of the selected outputs that's linked to the study and contains more details on the various approaches taken in this project. I would be happy to share this information with anyone interested. Moving on to the next example. Besides the processing of grapes, in the Western Cape province also have factories involved in the processing of apples and potatoes. So apples are processed into a wide range of products, but the factories that this study focused on was involved in the making of apple juice, while the potato factory was involved in the production of potato chips and other potato-based products for restaurants and the general consumer. Waste that is generated from fruit or vegetable processing has great potential for beneficiation. So many of these are quite high in cellulose and starch and sugars. And as such, when we performed a literature survey, it was clear that there are many different paths that can be followed to obtain value-added products from agri-industrial wastes, an overview of which can be seen in this diagram. With limited resources, however, it is impossible to focus on all potential avenues towards beneficiation. In this study, we therefore focused on the three wastes mentioned, which is apple, grape, and potato, and exploring two potential avenues, the production of enzymes and the production of bioethanol. So for this study, we focused on the fate of solid waste that is generated during the processing. For the potato processing, 3 sets of solid waste is generated after the removal of the skins, after cutting, and after the sorting for the final quality check prior to packaging. This waste either gets disposed of on landfill or it is sent to other farms for animal feed. During apple processing, for the preparation of apple juice, the solid waste that is generated is referred to as apple pomace, and this in turn consists of apple skins, which is quite rich in pectin and lignocellulose, apple pips, and apple stalks, which are also lignocellulose rich. Since the aim of this study was to determine the potential use of the different wastes in the production of enzymes, we first had to evaluate the key properties of the waste. So if there are any phenolics present, and that could potentially hinder the growth of our bacterial strains or fungal strains, and the presence of sugars, which can serve as a carbon source for growth. So the high phenolic content of the grape waste made it an unsuitable resource, while the apple and potato waste, with their low phenolics and high sugar content, being more optimal substrates. 3 different teams worked on this particular project. At the Rhodes University in South Africa, the aim was to look at the application of lignocellulose degrading enzymes that was produced by recombinant Aspergillus niger strains cultivated on the waste and tested in the study, and then basically looking at whether or not they can use it for the release of reducing sugars from lignocellulose rich substrates. At Stellenbosch University, the aim was to look at whether the waste can be used for bioethanol production, while our team at the CPUT evaluated the waste as potential sources for the production of lacase from Trametes pubescens and the application of the lacase then in biocatalysis. So the 3 recombinant Aspergillus niger strains developed by the team at Stellenbosch University and a control strain was used in the study, and they're all listed in the first table on the slide. The strains were cultivated in a fungal growth medium, containing various concentrations of apple pomace waste and potato processing waste. Optimization studies included determining the optimal temperature, pH, and waste load for the production of the enzymes endoglucanase, mannanase, and endoxylenase. Optimal production was observed in a 20% potato waste medium, even when potato wastewater instead of distilled water was used in the preparation of the growth medium. Similar levels of enzyme production was observed, as highlighted on this slide. These enzymes were then purified and applied in synergy studies for the degradation of model lignocellulose substrates. So synergy studies were performed at the Rhodes University. The enzymes were combined in different ratios to determine the optimal conditions for the release of reducing sugars from model substrates, such as beechwood xylan. And the highest degree of synergy was observed for the mannanase-xylanase combination, resulting in more than 2 milligrams per mole of reducing sugars released within a 30-minute incubation period. Synergy was also observed for the endoglucanase and mannanase, when a substrate mixture was used of carboxymethylcellulose, locust bean gum, and beechwood xylan, which is a bit more complex. But again, it resulted in release of approximately 2 milligrams per mole of reducing sugars, making these the production of these enzymes on the waste streams quite suitable for subsequent application. The commercially available bioethanol-producing yeast strain, ethanol red, was used or cultivated in the presence of various apple and potato waste by the team at Stellenbosch University. The optimal release of ethanol occurred after 70 hours of fermentation in a growth medium prepared from a starch-rich wastewater, reaching nearly 100 grams per liter of ethanol production, while a level of 20 grams per liter was obtained when a 20% solid potato waste was used. Calculating the theoretical yield based on the dry weight of substrate used, it showed that 200 grams per liter of potato solids yield an approximate amount of 30.6 grams per liter of ethanol, making potato waste a very good potential waste stream for bioethanol production. The fungal strain, Trametes pubescens, was cultivated in the presence of the 3 waste streams tested in the study by our team at the CPUT. After 3 days of cultivation, 3.5 units per mole of lacase activity was detected in a 50% pressed apple pomace growth medium, while 2.5 units per mole was detected after 7 days of growth in a 20% potato waste medium. The high level of phenolics in the grape waste stream greatly inhibited the growth of the fungal strain and therefore did not produce any enzyme. But when the 20% potato waste were supplemented with the red grape waste as an inducer, the addition of low concentrations of the red grape waste, up to 5%, enhanced the lacase production by Trametes pubescens in both types of potato waste, with more than 4 units per mole with the red grape waste compared to approximately 2 units per mole when no red grape waste was added. Furthermore, while maximum lacase production occurred on day 7 in the potato waste alone, as you can see here, production when red grape waste was added was not only greater, but also occurred earlier at days 3 to 5. So this waste stream can therefore be used as an enzyme production inducer. In a previous study, our team showed that the dimerization of ferulic acid, a phenolic that is found in a range of fruits and vegetables, can result in products that exhibit a higher antioxidant potential compared to the starting material. We therefore tested whether the lacase produced in this study could be applied in this particular biocatalysis reaction. The crude lacase obtained from cultivation in potato waste was applied in this bioconversion. This was then compared to the purified lacase under the same conditions. Analysis by thin layer chromatography, or TLC, as can be seen here, revealed that similar products were obtained irrespective of whether a purified Trametes pubescent lacase was used, or whether the supernatant from our proof of concept airlift or bubble reactors were used. Staining of the TLC plate with DPPH, which can be used to detect antioxidant activity linked to hydrogen ion donation, highlighted the antioxidant activity of the products that are produced, as seen by the white spots against the pink-purple background. So this study ultimately allowed our team to develop an integrated bioremediation and beneficiation proposal for bio-based waste streams, where value-added products such as enzymes, antioxidants, and bioethanol can be produced for application in a circular economy. Here are some of the outputs that were generated from this study. And again, for more details, please feel free to consult these publications or contact me if you're interested in knowing more about the study. And then my final example, I will focus on our most recent work on the use of lignocellulose-rich waste, canola fines, for bioplastic production. So the South Africa's canola industry is rapidly expanding, and it's quite a lucrative sector, largely centered within our Western Cape province. This is driven by increased global demand, advancements in the seed cultivars, and growing consumer trend for healthy oils. The production has therefore significantly increased over the past decade. Canola is a successful winter crop in South Africa, but it can also be grown under irrigation in summer, and it basically plays a key role in crop rotation and supply of animal feed. So because it's produced all year round, it basically makes an ideal resource then to explore for biorefinery applications. Researchers in our institute have shown that high sugar yields can be achieved from canola fines by pre-treating the biomass with low concentrations of acid and steam at a biomass loading of 5%, followed by enzyme hydrolysis with viscozyme or cellulase mixtures. The high sugar yields after pretreatment then makes the canola fines a promising lignocellulosic feedstock for polyhydroxybutyrate or PHB production. PHB has become quite topical as a bioplastic that can be used to replace fossil-based non-biodegradable plastics. And luckily for us, actinomycetes are well known for their biosynthetic potential and have been reported previously to have the ability to produce the bioplastic PHB. In this study, 3 Gordonia species, 2 streptomycetes, a Micromonospora, and a Nocardia strain were used. And these strains were all isolated from diverse environments with the aim to see whether the environmental source may influence the ability of the strain to produce PHB. Optimal production of PHB in a defined medium, in this case, the International Streptomyces Project Medium 2, was observed for strain Nocardia humpkensis, which was isolated from soil collected from a riverbank, followed by the Gordonia strains. Interestingly, the lowest yields were observed for the streptomycete strains and the Micromonospora, but the study was the first to report on a Micromonospora strain with the ability to produce PHB, even if it was at low concentrations. Analysis of the genomes of the 3 Gordonia strains, the Nocardia humpkensis and Streptomyces strain HMT19, revealed that none of the core genes were located in defined operants, but all 3 genes required for PHB production are present, confirming the biosynthetic potential of the strains to produce PHB. It was interesting to see that Gordonia strain JC51, which showed potential when grown in ISP2, did not grow well and produce very low amounts of PHB in a synthetic canola fines hydrolysate. On the other hand, the Gordonia BS2 that did not grow well in ISP2 and produced low amounts of PHB in this particular medium grew very well and produced the highest amount of PHB of all the strains in the synthetic hydrolysate. However, the maximum growth and PHB production took a bit longer than with Gordonia strain BG1.3. And although the Gordonia strain JC51 utilized similar the salabaios in similar amounts to the other strains of Gordonia, it was not able to utilize arabinose and only utilize about 1% of the xylose in the synthetic hydrolysate. So obviously, at the moment, current optimization studies is focused on the two Gordonia strains, BS2 and BG1.3. And just for interest, the one was isolated from a lagoon, while the other was from a peatland. So in conclusion, for the winery wastewater treatment project, it can be concluded that the biological sand filtration system can provide a low-cost, energy-efficient, low-maintenance, and a sustainable means of treating cellar effluent at small wineries. And this project required a transdisciplinary approach involving academics from different disciplines and the farming community, ultimately leading to research uptake. And this is obviously what we all want to achieve with our research. For the bio-based waste streams project, we showed the various applications or abilities to either produce enzymes and show their application, and then also the production of bioethanol from the potato waste, making it very promising resources. The study then allowed for the design of a proposed integrated bioremediation and beneficiation process, promoting a circular economy. And then finally, for the canola fines project, we could see that the pre-treated canola fines contain relevant sugars for the growth of actinomycete strains with the ability to produce the bioplastic PHB. And this approach theoretically could then provide both environmental and economic benefits in support of a circular economy. With that, I wish to thank everyone involved in the research presented, as well as the various funders that made this research possible, especially the Water Research Commission of South Africa. And thank you all for listening. If anyone is interested in learning more about these projects and the current work, please feel free to contact me.